And today I'm really happy to welcome Nick Grebe from Occidental College. Nick started this fall as an assistant professor in psychology at Occidental, so is now here in LA with us, and it's a real pleasure to have you here. And we're going to hear about his research on lemurs, mountain gorillas, and people. I think there's a little bit.
Yeah, we'll mix it in there as well.
So please join me in welcoming
All right. Thanks, Clark, for inviting me. I'm very pleased to contribute a bit to this very storied speaker series. Hopefully you're ready to hear about some unusual primates and what they might be able to tell us about ourselves and models of social evolution. Okay, so I won't leave you in suspense. I'll start by helping those out who have no idea what a snark is and wondering why it's in the title of the talk. So, but maybe some of you already know the Lewis Carroll short story, The Hunting of the Snark. Lewis Carroll is the same guy who wrote Alice in Wonderland. So this story is kind of simultaneously confusing and absurd, a little whimsical, and some part grim. The plot, as much as you can call it one, concerns a group of characters, including an anthropomorphic beaver, who travel to a strange island and go hunting for mysterious creatures called snarks. Most snarks are good quarry, but some are in fact boojums in disguise. And a boojum is a dangerous variant of a snark that will make a hunter disappear at the very sight of them. So Carroll's book had all these really cool, creepy woodcut illustrations. I like them, so I've sprinkled them throughout my slides. You never actually see what a snark looks like in the book, but there is this ominous illustration towards the end where you can see what looks like the outline of a face. If you kind of squint and look closely, you can kind of see the nose there. It's like a rightward-facing face. I'm not sure if this is supposed to be one of the characters, the baker, who at the end of the story encounters a boojum and then vanishes. Maybe this is supposed to be the boojum itself. Either way, the point is that hunting a boojum is something to be avoided, and it's hard to avoid times. You can be tricked easily. So it's been a long time, 75 years now, since this story was first used as an allegory for the research process. I always kind of assumed, because I had read it early in grad school and was taught it, that the 1950 paper by Frank Beach called The Snark Was a Boojum was something kind of well known in scientific circles, but I've learned that isn't necessarily the case, so I'll just talk about it real quick. Frank Beach was one of the founders of the field of behavioral endocrinology, right? The field that studies how hormones affect behavior and vice versa. And by the time 1950 had rolled around, already the field had grown quite a bit in its technical complexity. It's one of the older, I'd say, subfields of biology. This worried Beach, however, because as he put it, the most popular journals at the time might as well have been called the Journals of Rat Studies, due to scholars' reliance on lab rats for experiments. And this was, kind of the heyday of behaviorism in psychology. It became a popular perspective to think that what was most possible or most desirable for scientific and technical progress was having a tractable animal model, right? Principles of behavior are largely universal across species, so went the tenets of sort of radical behaviorism. And on the left is Beach's response to that position, right? So the comparative psychologist was hunting a snark known as animal behavior. His techniques were different as the baker, but he came to the same unhappy end because the snark actually proved to be a boojum. Instead of animals in the generic sense, he only found one animal, and thereupon the comparative psychologist suddenly and softly vanished away, poetically put. I also included this figure from Beach's paper. I think it's kind of funny that he had basically a political cartoon commission to demonstrate his point, but I think it demonstrates it nicely, right? A group of scientists following the rat off in some unknown direction, just sort of slavishly following whatever the rat has to say. Okay, so comparative psychologists and comparative neuroscientists have made a lot of hay out of this metaphor. There have been multiple follow-ups to the classic 1950 paper, basically like revisiting the question, are we hunting snarks or are we actually hunting boojums? And the general consensus is that, well, there are a ton of reasons to do the former, but the latter is always kind of a temptation to perennially guard against. There are, sort of like concrete victories that we can point to from a broad comparative approach, right? Like in my neuroscience class, I teach about how things like the ionic basis of the action potential, the discovery of adult neurogenesis, or like the cellular basis of learning and memory came not initially from rats, but from creatures like squid or canaries or sea slugs. But often it's hard to predict what sort of applied value is going to come from basic research, right? So there's a broader range of reasons to take a broader comparative approach. It could be to determine how an organism has solved a unique environmental problem. That could be one reason we want to take this approach. We might be interested in testing the generalizability of what's been found in one organism or a group of organisms. We might have specific hypotheses in mind that we want to test or try to falsify about how physiology should affect behavior in certain ecological contexts. And as evolutionary scientists, we probably are interested in when biological mechanisms might have evolved, so we want to do a broad comparative approach for phylogenetic analyses. We might want to see if similar ecological or social conditions led distantly related organisms to converge on sort of similar mechanisms. Or it could just be because the species is unusual, or for the sheer love of the species, as Adkins put it. Adkins Reagan, rather. So I list all these out. I took the time to because I think each of them, to some extent, kind of captures what motivated me to study the biology of monogamy in these groups of primates. So today I'm going to run through what I'm calling two tales of snark hunting, or at least I hope it's snark hunting. The topic for these tales of each is the biology and evolution of monogamy. The first is a more completed line of work concerning the role of oxytocin in interspecific mating variation in lemurs. And then the second is more in the earlier stages of planning and pilot data analyses on the use of mountain gorillas as a potential underexplored model for the evolution of pair bond-like relationships in the ape lineage. Okay, so to talk about this topic, it's important to kind of first lay out some fundamental definitional issues. Monogamy is actually a pretty complicated construct to define because it encompasses a bunch of traits that sometimes, but not always, go together, and often these traits are conflated, not just by people outside the field, but from researchers themselves. So part of monogamy is what we call pair living. It's exactly what it sounds like, right? Mating pairs live and associate with each other instead of living on their own or living in larger social groups. Another part is being pair bonded, which is sort of the behavioral or psychological aspect of monogamy. Pair bonded animals are, clearly attached to one another. They are upset when they're separated. They have these pair-specific behavioral rituals. They tend to be more interested in spending time with each other than other individuals, things like that. There's the element of sexual or genetic monogamy, which is that, the pair produce offspring only with each other and no one else. And then finally, there's the element of biparental care. Frequently, monogamous species mate together and also raise offspring together. And it's thought that this parental care, this biparental care, sort of stems as something downstream from other aspects of monogamy. And so all of these features are kind of sometimes subsumed under this umbrella of monogamy, but not all species that people might call monogamous have all of these features together. And that's an issue that we'll return to later. But for now, you can just think of these features as sort of characteristics that often overlap, and the degree to which they overlap really depends on the model in question. Okay, so let's take a step back. Why do species vary in their mating systems anyway? Why are some species generally monogamous, generally polygynous, generally promiscuous, whatever that might be defined by in terms of those features? Well, so between broad taxonomic levels, biologists often talk about life history constraints, such as whether animals give birth to live young or not. Within zooming in, more closely related taxonomic groups, ultimate explanations often appeal to differences in environmental niches that are occupied by species that have these different mating systems. But if we're talking about proximate mechanisms that explain this variation, the comparative neuroscience literature provides some really helpful context to this broad question. So within the vast web of organismal diversity that characterizes all these different taxonomic groups, invertebrates, for instance, one unifying observation is the conservation of what's been called a social decision-making network. That's a set of brain structures that mediate information about the salience of a social stimulus into an adaptive behavioral output. What this means is that much of the raw sort of neural ingredients that are proximally responsible for adaptive social behavior are more or less conserved across vertebrates. But these raw ingredients are arranged differentially in networks to such an extent that it can manifest in, say, completely opposite modes of mating and rearing offspring. So that's kind of the question that motivated this first study. Are there neural factors that in these networks, in these specific areas of the brain that we know from past research tend to be related to differences in mating system, that can explain interspecific differences in monogamous pair bonding. Okay, so within this research question, scientists have focused on a pair of hormones pretty intensively. That would be the 9 amino acid peptides oxytocin and vasopressin. These are two closely related hormones, actually originally resulting from a gene duplication event. They are produced in the hypothalamus of all mammals, and they're homologous molecules in other vertebrate animal groups. But in mammals specifically, oxytocin is really important for the processes of giving birth and nursing offspring. The idea is that this hormone has then been co-opted in multiple stages to perform new functions, perhaps to mediate the process of maternal-infant social bonding, and then in some cases to mediate the bond between mating partners. So scientists first developed these ideas from looking at different species of voles that are shown up on the screen here. So it turns out that some vole species, like the prairie voles here, form pair bonds and raise young together, but others, like for instance the meadow voles on the right, don't. So scientists wondered whether that could be explained by oxytocin working differently in the two species. And on this slide are a couple of representative findings that are really seminal for this line of research. So Insel and colleagues first in one study back in the early 90s looked inside the brains of voles and found that oxytocin and vasopressin go to different parts of the brain in pair bonding voles, shown here in the green on the left, compared to non-pair bonding meadow voles in orange on the right. You can probably see the differences yourself from just eyeballing it. You know, these are very large effect sizes for the species comparisons. And furthermore, future work revealed that non-monogamous voles can be made to look like monogamous voles by altering the vasopressin receptor system here. So this figure here in the bottom panel, this AAV treatment, this is basically inserting vasopressin receptors into a non-monogamous meadow vole. And what you see is, yes, not only does it look more like a prairie vole brain, but in fact the non-monogamous meadow voles with this vasopressin receptor treatment behave a lot more like monogamous prairie voles in terms of forming pair bonds. So these were, and still are, frankly, really exciting findings because they strongly suggest that, at least in a rodent model, oxytocin and vasopressin acting within some of these circuits in the social decision-making network are necessary to form a pair bond. So this is the basis for statements like this one in the bottom right that came from a high-profile review of this kind of research saying that, well, differential regulation of neuropeptide receptors may explain species differences in the ability to form pair bonds. Okay, so this was first I want to mention a real victory for the comparative approach, right? These insights would have been possible if there wasn't this comparison of these different vole species. And being rodents, voles were and are really well suited to certain kinds of designs and certain kinds of research questions. But how can we assure ourselves that we're not chasing a boogem or just replacing a fixation on rats with a fixation on voles or something like that? If we want to understand ourselves, for instance, it might be a good idea to look at our primate cousins. And indeed, there are some nuances to research in this domain that make it, I think, helpful to consider why studying primates might be really, really useful and show limitations also of relying on rodent models. So one aspect is just biochemical. While oxytocin appears very specific to its receptors in rodents, for whatever reason, in primates, there's an extensive amount of cross-binding between oxytocin and vasopressin receptors, right? So that's what this figure from Freeman et al. Shows, is that there's some sort of promiscuous binding of each of these hormones to each of the receptors to varying degrees. So we need, among other things, special methods to accurately map both receptors. And more generally, when we have looked at the limited number of species thus far that have had their oxytocin receptors mapped, the map looks quite different from the primate study to date and really any rodent. So this suggests that there might be a ceiling or an upper limit to any sort of translational value of any rodent model, no matter how elegant. So taking these complexities into account, how might we pick a useful model for examining oxytocin's functions within primates? Of course, within the order, there's a ton of diversity in social systems. All the groups you see in red on the figure on the right here have some elements of monogamy, even if not all of them. Jeff French and his group call them monogamish, which is kind of a catchy way to put it. But of course, there are many more differences between these species that are in red, besides the fact that they just share a sort of rough designation of mating system. primate groups have evolved to, of course, live very different lives in terms of morphology, diet, preferred habitat, and so on. So if we really want to compare apples to apples as best we can and be able to isolate specific hormonal factors that might explain interspecific social differences, it would be really nice to have a natural experiment that's something sort of akin to the voles, right? A situation where, for example, closely related primates that are within the same genus have very different mating systems. And that's where lemurs come in. So since lemurs arrived in Madagascar, they've spread out and adapted to very different environments, leading them to develop, among other things, a wide range of social systems. So within the eulemur genus specifically, which consists of 12 species that live all across the island, there's a remarkable diversity of mating systems. Some live in large social groups and mate promiscuously, like the black lemurs and the blue-eyed black lemurs that I just put up there. Others form pair bonds and live in small family groups, like the mongoose lemurs or the red-bellied lemurs shown at the bottom there. So this arrangement where eulemur species show these opposing social or mating systems is pretty much unparalleled in the primate order. You can't find these different mating systems in such a close phylogenetic space otherwise. And so this natural experiment was the basis for both of our studies. In the first study, what we did is we compared the neuroanatomy of monogamous and non-monogamous eulemur species, mapping out the locations of both oxytocin and vasopressin receptors across the entire brain. We didn't know what we'd find. Obviously, lemurs are not a popular model for neuroanatomy or for neuroscience, so we basically were just trying to map the entire brain and see what we found. All of our specimens, I should note, came from animals that lived at the Duke Lemur Center and died of natural causes, right? So none of the animals died for this study. This was all post-mortem brain tissue that was banked by the Lemur Center that we purchased and used. Our team used 12 specimens, 11 hemispheres, 1 whole brain, from 7 closely related species, and you can see the breakdown of sex, species, and mating system in this figure here on the right. Okay, so to show some pictures, here are some figures depicting some representative results, at least. So first here is the brain of a monogamous red-bellied lemur in a coronal plane, like this, going from the front of the brain to the back across the 2 panels, so front being up here and then going to the back, like that. Dark areas in columns A and D mean more oxytocin receptors in that region. Dark in B and E means more vasopressin receptors, and then C and E is a counter stain, because we had to do a lot of looking at these areas under a microscope. There is no lemur brain atlas, so there was a lot of us trying to figure out what exactly we were looking at with some of these specimens. So some of the sites of binding for both those hormone receptors were expected based off past studies of non-human primates, but there were also a number of surprises. For instance, binding in the nucleus accumbens, and then the medial geniculate nucleus, where is that? There. Actually is more reminiscent of findings from rodents than any of the previous work that had been done in primates. So lemurs, in some aspects, we think, appear to exhibit a mix of binding patterns for these hormone receptors that's sort of between the taxonomic groups of rodents and primates. And next, here's the brain of a non-monogamous black lemur. So there were some differences from the red-bellied lemur brain, and if you look very closely, you might be able to see some of them visually, but you're not supposed to be able to see the sort of stark differences like you saw with the vole brains, and that's why I put them up on the screen, right? The differences that we found were pretty inconsistent. When they did exist, they were much smaller than the corresponding differences in voles. And most importantly, they weren't usually observed in the areas that are hypothesized to be key from the established models that were from rodents or other non-human primates. Basically, all the differences that we did observe were swamped by individual variation within the brains of lemurs from the same species, actually. So here's a figure. We did actually run some statistics, and this summarizes results across the entire brain. Warmer colors correspond to more dense binding in that region, and you can see that monogamous versus non-monogamous are the 2 columns, and then we just broke it down by various sort of larger organizational parts of the brain. So again, the original papers from these vole models showed a distinct, very distinct split between monogamous and non-monogamous brains, huge effect sizes. But that's not what we found in eulemur. You can see a few areas where the average binding differs between mating systems, in a few cases significantly, but these weren't found in putative regions of the pair bonding circuit. Okay, so here it looks like lemurs exhibit a mix of primate-like and rodent-like oxytocin and vasopressin receptor distributions, right? Shared areas with primates, I think, do support the idea that oxytocin and vasopressin are strongly influenced by visual processing in primates compared to olfactory processing in rodents, sort of one of the things that distinguishes these two mammalian groups. But at the same time, other locations are more consistent with olfactory-oriented rodents, which sort of makes sense if you know how lemurs and other strepsirrhine primates are sort of olfactorily oriented. And there actually is a little bit of evidence, oddly enough, from I think galagos, where they looked at these oxytocin receptor distributions. In galagos, it seems pretty similar to what we observed in lemurs. So this is all consistent, we think, with lemurs occupying an intermediate evolutionary niche between rodents and haplorhine primates, which they do in a few different ways. But on the other end, there's little to no evidence that differences in oxytocin or vasopressin receptor distributions are distinguishing monogamous from non-monogamous lemurs. But as an interesting postscript to this study, grad student Ali Schrock, she actually has been building on this data set for the past couple of years. And what she does, she actually looked at systematic variation as a function of dominant system rather than mating systems. So what she found is that, well, there was more systematic variation in some regions of the social decision-making network. Here we're talking about different nuclei of the amygdala. But again, it wasn't that these differences were predicted by mating system. They were much better predicted by dominant system. Something that I'm totally glossing over, but is another interesting wrinkle about lemurs, is that they have a lot of variation between species in terms of how strongly female dominant they are. So most of them are strongly female dominant, but there are also a decent number of species that are more egalitarian or co-dominant. And what Ali found was that, well, it turns out that the co-dominant species have greater density of oxytocin receptors in different regions of the nuclei compared to the female dominant species. So it could be, perhaps, that neuropeptides are better predictors of dominant structure in this taxonomic group than mating structure. Okay, so that's a bit about the neuroanatomy, but what about behavior, right? We're interested in that. What are maybe, for instance, the consequences of modifying oxytocin activity in the central nervous system within the social decision-making network? So that's what we sought to do here. So like with the neuroanatomy, this has been explored extensively in rodents, but I'm not going to talk about that because unlike the neuroanatomy, this is a research question that already has received a good bit of attention in the primate literature. And in this case, at the time that we were working on this project, almost all, not all, but almost all of the evidence base consisted of studies from New World monkeys that form these monogamous pair bonds, marmosets and tamarins. So here's, a list of some of the studies that were available at the time when we were designing this project, assessing the effects of either administering oxytocin, OT, or administering an oxytocin receptor antagonist, the OTA. It was nice to have these studies, but when I was digging into what these studies found and trying to figure out how we could develop predictions for our projects, what I realized is that really the findings are kind of all over the place, right? So we can look at the second column here, just, okay, well, were the behavioral effects of modifying oxytocin significant? And the answer is, well, sometimes yes, sometimes no. It seems to depend on the study or the outcome measure looked at, right? Obviously, the significant effects were the things that were emphasized, but when you dig into these papers, you see it's a little more heterogeneous. And then, just beyond sort of significant main effects, sometimes papers examined whether the effects of these hormonal manipulations differed between males and females. Again, sometimes yes, sometimes no, sometimes it's not reported, sometimes I couldn't make sense of whether it was different between the sexes or not. And then lastly, in the final column, one possibility, right, is that administering oxytocin or a blockade to a monkey affects their behavior, which would be an actor effect, and or, it's not mutually exclusive, administering that same treatment to a monkey could affect that monkey's partner's behavior, right? So that would be a partner effect. And again, it varies depending on whether actor effects were examined, partner effects, or both. Okay, so what we were trying to do was, from this evidence base, which, to be clear, was like absolutely key for our study. Like so much of the methods and so much of what we did built upon these studies, so I'm not trashing them by any means, but it was a bit of a challenge to extract what were sort of the key predictions and findings from these past studies, while at the same time in our design, leaving some room to explore these sort of auxiliary findings or sort of inconsistently reported findings. And so what we did is we had a pre-registered study of oxytocin receptor blockade in pairs of monogamous and non-monogamous eulemur. We only did the oxytocin receptor antagonist for reasons I can get into. But what we did for this study is we had a set of confirmatory predictions in our pre-registration, right? And so the confirmatory predictions were, I think, pretty straightforward. They stem directly from the literature on that past slide that I showed you. So first, we expected to see baseline differences in what we're operationalizing as affiliative behaviors. So that would be spending time in close proximity, grooming, and for lemurs especially, scent marking each other. They do a lot of that. And then second, we predicted that, okay, well, that's the control condition for number one. And then for number two, when we would actually block oxytocin receptors, the antagonist should decrease the overall rates of these affiliative behaviors, but only in monogamous lemur pairs. We didn't expect an effect in the non-monogamous pairs. So here's kind of a representative finding from one of those studies. You can see that the white bar, that's oxytocin plus. That's when you administer oxytocin. Gray bar is control, and then the black bar is oxytocin minus the antagonist. So you can see that the rates of those affiliative behaviors tended to decrease with the antagonist treatment. But we didn't want to just administer the drug and then see what happens. That was part of it, but we also wanted to sort of introduce a challenge or something sort of salient to the lemurs after either getting the drug or not. And so what we did is we did what we call a bioassay, where we put some dowels into their enclosures, and the dowels either have a control scent, it's like peppermint oil rubbed on them, or they have the scent that comes from lemur scent gland secretion rubbed onto it. And lemurs love that, right? They're already very interested in scent marking anything you put in their enclosure, but if it has, say, the scent of a potential reproductive competitor, which some of the dowels did, they are very interested in it, obviously. So we also predicted that the bioassay responses, scent marking, affiliation, after smelling a reproductive competitor, stuff like that, would be affected by the oxytocin receptor antagonist, but again, only in the monogamous pairs. And also, I want to note, this doesn't preclude us from examining any exploratory comparisons regarding, say, sex-specific effects or other interactions, just that these weren't the core predictions from past research. And also these auxiliary sort of findings were tested in really flexible ways. So they weren't part of our pre-registration, even though we wanted to and did end up looking at them. Okay, so videos are always fun. All right, so all the research that we did was non-invasive. Lemurs are primates, and they're all endangered or critically endangered. So there was no restraining, no injections, no anything like that. But we had to, we still wanted to modify their hormonal systems, right? Well, luckily for the oxytocin receptor antagonist, there is a drug that can be orally administered, right? So we had to get creative because we had to get the lemurs to eat the drug. They are sometimes very finicky eaters, and the drug tastes like medicine. It tastes bitter. So we had to trick them into eating it, getting the full dose. And sometimes it worked pretty well. So here's an example of, I think, it working pretty well. This is, who's that? That's Terry's. You'll notice this is a ring-tailed lemur, not a ewe-lemur. The reason was because we wanted to build up our sample size of the non-monogamous lemur pairs, so we added some ring-tailed lemurs in there. So Terry's is happy as a clam. There's medicine in there, but he doesn't even know it because it's mixed in with banana and grapes and primate chow and other stuff like that. So he got all the medicine there. Perfect. Star student. And then here's Terry's later on. So he was doing so well that we decided to just leave the medicine in there inside the food and see if he would eat it that way. So this is him in a different trial. You see a little head shake there. He keeps going. He powers through. Gets a big dose of it there and does a big head shake. That's always fun because then they fling food that has medicine inside of it all over their enclosure. So I had to go all over the place and get these little dots of food and collect them together and cut it with more grapes or something like that to get them to eat it. But luckily he still continued to eat it. And then other times it really just didn't work well at all. So here's, this is actually Terry's enclosure mate, Persephone. And Persephone was not having it. So she's up there. You can see right away she starts reacting to the medicine. Tons of shakes. I think we even get a gag here. gag. And then Aaron says, okay, well, we're just going to give Persephone a treat. This had no drug in it. She's like, okay, will you just eat a grape? No, she's suspicious now. So we had to eventually give up on Persephone. She got very, very suspicious of us and would not take anything from us, and obviously we don't want that. So sometimes animals got dropped from the study, but luckily the vast majority of the time we were able to get them to take all the drug, and we were able to retain them in the study. Oh, and in case you're curious what the bioassay looks like, so here's a bioassay with a pair of red-bellied lemurs. You will see 2 dowels here in a second, and you'll see kind of how they act. So that's the male there with more of the white on his mask. And then the female will pop up here in a second. Okay, so 2 dowels, left and right. Which one do you think was the one that had the lemur scent on it? The right, yeah. They're much more interested in that one on the right than the left. They kind of do a cursory checkout of the one on the left, but obviously they spend much, much more time on the scented dowel. Didn't see any actual scent marking behavior there, but they'll do that. They'll rub their wrists over it. They'll rub their genitals over it. I mean, they love it. They can't get enough of scent marking stuff that you put in their enclosure.
One quick question.
Yeah.
So the monogamous pairs were put in and exposed to the dowels as a pair.
Mm-hmm.
The non-monogamous, were they also in mixed sex pairs?
They were, yeah. So all of the non-monogamous species were housed as opposite sex pairs, and were also exposed to the bioassay in the exact same way. Yeah, it was completely, all the factors were completely crossed in this experiment. Okay, so what do we find? Well, let's start with the non-monogamous, which is the cluster of bars on the left there. Blocking oxytocin receptors, whether it was in just the male, which would be the, the yellow bar, just in the female, which would be the green bar, or both, which is kind of the salmon colored or the pink bar, had no effect on behavior. And that's what we anticipated, right? We didn't expect that the oxytocin receptor antagonist would affect their behavior, and it didn't seem to. For the monogamous species, it was a little interesting, right? So blocking oxytocin receptors in either the male or the female, right? We had all 4 of these conditions. If it was just one or the other member of the pair, it didn't seem to have any effect on their behavior. However, that pink colored bar, that's the one where they both got the oxytocin receptor antagonist at the same time. There, that did lead to a significant behavioral change, both from the control condition and either of the single member oxytocin receptor antagonist condition. So that did seem to have an effect. Interesting and unexpected, actually. Right, so here we found pretty weak or inconsistent evidence for our confirmatory predictions, the ones that I laid out, right? You also might have been able to tell from this last slide that the non-monogamous in the control condition actually had slightly higher rates of affiliative behavior than the monogamous in the control condition, looking at those 2 blue bars there. It wasn't significant, but went in the wrong direction, or the unexpected direction at least. So not much support for these confirmatory predictions, but some suggestion of a sort of exploratory synergistic, we're calling receptor blockade effect, right? When you administer it to both members of a monogamous pair at the same time. So it seems like hormonal disruption in one member of a pair doesn't reliably affect dyadic behavior, but simultaneous disruption does, or at least it might. We suggest, as a speculative explanation, that there could be some degree of partner compensation here. You can imagine that if blocking oxytocin receptors sort of decreases, say, the reward that an individual perceives from initiating some sort of affiliative behavior with their partner, well, the other partner can just initiate more often to sort of make up some of that slack. And that could be one reason why we didn't see significant differences in either the single partner conditions. But if you're blocking oxytocin receptors simultaneously in both, there's no one to initiate, right? And so the rates of these behaviors do decrease. Again, this is speculative. This is one of the exploratory sort of analyses that we looked at. It's plausible, but I would very much like to see this get tested in some other species. Because surprisingly, for all the studies that have been done on the oxytocin receptor antagonists in other primate species, no one else has looked at if there's a difference between it blocking oxytocin receptors in just one member of the pair versus both at the same time. None of those studies did that. Not sure why, but it would be really interesting to see what happens if they did. So, and just, as a food for thought, I fully admit that this was not part of our confirmatory predictions, so how much stock should one put in these findings that are plausible, but at the same time exploratory? I think that's an open question. Again, I would really like to see whether sort of similar things emerge when looking at, for instance, the same sort of paradigm in marmosets or tamarins. Okay, let's switch gears now. Okay, so as I mentioned earlier, this tale is one that's sort of just beginning. Unlike the lemur research, it's more in the planning stage, more in the pilot data analysis stage. You'll kind of see that there's a lot of different threads that are sort of trying to be wove together, but there's also some hanging ones. Happy to talk about any of this further. Hopefully in the Q&A session we can talk about any of this, but particularly this part on this project that we're working on now with mountain gorillas. Okay, so the basis for this project comes from questioning why New World monkeys should be the main primate model for human monogamy. Obviously, if we take a TD monkey, for instance, they share the pair bonding and the biparental care aspect with us, which is great. But in some other ways, they're quite different. We're quite different from a TD monkey. TD monkeys are sexually monomorphic. They live in small groups that consist almost entirely of immediate family members. Marmosets and tamarins sometimes have polyandrous mating systems and social configurations, but usually small family groups is what you see in the models that have been used for primate monogamy. And there's a focus on limited calorically dense foods, tree gums, fruits, things like that. Compare that to us, we are moderately sexually dimorphic. I know there's tons of debate about how dimorphic we actually are. Depends on the feature in question, of course, but I think that it's probably fair to say that we are, on average, more sexually dimorphic than a TD monkey. We live in complex, multi-level societies that combine kin and non-kin, which is a bit of contrast from, say, a TD monkey. Of course, we have basically every subsistence pattern you can imagine, right? So in some ways, right, we are very, very different from the models that have been dominant thus far in studying monogamy and the biology of monogamy. Fully admit that this argument also kind of takes away some of the comparative power of what I was just talking about with the lemurs, right? But I guess what I think of it as is like there are two different angles for approaching this problem, right? One angle with the lemurs is taking, right, like a very small phylogenetic space and seeing if you can isolate specific factors that explain differences in this relatively narrow space. That allows you, I think, to have some degree of nice control. And then the other angle is to take something that's a lot more broad and to look at, for instance, a close human relative and to measure a number of different features that have been underexplored thus far and see if possibly this model has some promise for providing new light to a question that has been discussed for a long time, but new data and new observations from these species are sort of calling into question the dominant models of social evolution, which I think work in the mountain gorillas do that. Okay, so I'm going to argue that despite not being a classic model for monogamy by any means, mountain gorillas are nonetheless a very promising and interesting model to study based on what we know about their behavior and their sociality. And I put this figure back up that I already showed you to remind you to keep an eye toward these different features that I talked about at the beginning of the talk, these different features that are sort of subsumed under this umbrella of monogamy but are dissociable and don't always overlap necessarily. Okay, so I'm going to tell you a bit about the research that's been done at the Dian Fossey Gorilla Fund. It speaks to the promise of mountain gorillas as a model. It has led to the questions that I'm interested in exploring next. I've been involved in some of it over the last few years as a postdoc and then now as a research partner. But much of it is that I'm going to talk about is long-term data that has been organized and led by the people you see, among others, on this slide, including Stacy Rosenbaum on the upper left, who some of you might recognize as a graduate of the Anthro program here. Okay, so first observation, mountain gorillas have a degree of social flexibility that you don't see often, including not in other gorilla species. So the classic gorilla group that you're probably imagining of the silverback, one or more females and their kids, is a configuration you see if you just count the number of groups across the history of the FOSSI fund, it happens about 60% of the time, but that means there's also a significant number of groups that live in multi-male, multi-female groups. And this is fluid, right? There are some eras where most of the groups are single male, some eras where most of the groups are multi-male. It just so happens that a lot of the long-term data that I'll talk about, when we had the sort of the behaviors that we were most interested in, most of the groups were multi-male, but that wasn't necessarily representative of their entire known history. And group composition is fluid to some degree. So females will transfer during intergroup interactions. Males may strike out on their own when they reach maturity. They might stay in their natal groups. It seems to be about 50-50. It's clearly a system that has a degree of wiggle room. And yet, at the same time, even though you have this flexibility, and in multi-male groups, many males become fathers in them, dominant males are clearly overrepresented, right? There's strong reproductive skew. So this figure on the right is showing the reproductive history of some of the longer tenured males in multi-male groups. Their paternities are the colored dots, right? And then paternities from some other male besides them is in either a gray or a white dot. And you can see that, yeah, there's a substantial minority of gray or white dots for each of these males, but most of them are the colored dots. The paternities are from the dominant male in the group. Okay, and next, even though mountain gorillas have kin exposure to both maternal and paternal relatives and full siblings because of this social flexibility, something that sort of is like humans and is an interesting sort of comparative model for human kin discrimination, it looks like mountain gorillas' mechanisms or abilities to distinguish and favor paternal kin in particular are pretty spotty. I don't want to say absent, but I'm going to say spotty. So that's what this figure here on the right shows from a paper that we published a couple of years ago. It looks like, at least for some, the paternal kin are not very distinguishable from non-kin in terms of the patterns of individuals choosing to affiliate with any of the other gorillas in their group. So there are a number of potential interpretations of this pattern, but one that we put forward is that transitions from strict polygyny in mountain gorillas might be relatively recent, right? Perhaps their behavior hasn't quite had time to catch up to what is the reality of mountain gorilla social structure today, which is a mixture of polygyny and then multi-male, multi-female groups. Okay, so that's kind of one end. But I think what's surprising, based off of this information about kin discrimination and about mating patterns, mountain gorilla males are involved dads. They're highly oriented towards offspring, that includes genetic offspring, but also just they act as sort of social fathers to infants in the group that they're not genetically related to. And this infant care tends to come more in the form of things like play, protection from infanticide, the occasional adoption of infants, which is interesting. It's not direct provisioning, but it's a lot of investment. And it, again, happens sometimes towards kids that are not genetically theirs. And also, interestingly enough, the amount to which males engage in this kind of paternal care predicts their future reproductive success. So those observations together suggest that perhaps infant care in mountain gorillas functions to some extent as mating effort rather than parenting effort. And you might note that what I'm describing with this is paternal care, but it exists without social monogamy or genetic monogamy, right? And that really challenges and contradicts the conclusions of some other pretty high-profile comparative analyses of primate social evolution. You know, this one from a PNAS paper about 10 years ago says that, well, one of their main conclusions is that paternal care evolves only after a switch to social monogamy, not in polygynous systems. It's obviously not the case here. So what that suggests is that, well, the existing models of primate social evolution maybe need to be revisited, and studying mountain gorillas in more depth might help us sort of modify our ideas about how monogamy might have evolved in the primate order, and including it in humans. Okay, and additionally, some of the pilot analyses that we've been working on suggest that there are some aspects of mating bonds in mountain gorillas that resemble monogamous pair bonds. So when reading through some really old observational papers, the mountain gorillas at Karasoke have been followed for like 50 years now. And so there's a bunch of these cool older papers with these interesting anecdotal observations. And one thing that popped out a lot was this claim that male-female relationships between unrelated individuals were among the strongest class of social bonds in mountain gorilla society. So we started looking at whether this was reflected in any of the data that we had immediate access to in the long-term data set. And indeed, we do see some signs that unrelated male-female dyads are socially attuned to one another, not just in the period surrounding mating. So this figure here on the right is not just the period surrounding mating, it's actually just all of the observational periods that we have. So what this shows is rates of social staring by dyad class. And social staring is this behavior where an individual will approach another and get really close. Like the cutoff we use is within a half a meter. And they will look at each other. And it's usually not direct eye contact, but it's like they're staring at a different part of the other's face or something like that. And it's an affiliative behavior that you see a lot in various classes of dyads, including, surprisingly enough, unrelated male-female pairs, right? So this is an important affiliative behavior in gorillas, and we see actually quite high rates of it in unrelated adult males and adult females. And again, this isn't just something that's in the immediate period surrounding mating, right? It goes up during that period, but this is something that seems to happen consistently. And finally, we also see some hints of differentiation of social relationships, right, around mating periods. So this figure shows a couple things. So two things are simultaneously true about what we know thus far. Females have active preferences for particular males. So what this is showing in panel A is that Africa has a preference for silverback 1, then silverback 4, and then not really for 2 or 3, whereas female Ashema has a pretty strong preference for silverback 2 and then none of the others. Right, so there's a degree of differentiation. At the same time, though, if you just look at all the females together, certain males are disproportionately preferred on average. It's usually, but not always, the dominant male in the group. So those two things coexist with each other. But this pattern of proximity that's depicted in panel A, well, it predicts rates of copulation during a mating period following it, right? So there are some consequences, we think, to this differentiation of how much time individual pairs are spending around each other. But we don't know yet if this generalizes outside of the immediate mating period, and like with the social staring, we'd want to see that, of course, to be able to say, like, okay, well, we have some characteristics here of like what we might call proto-pair bonding, or if you know the baboon literature that Joan Silk has worked on, primary associations is one of the terms she uses and that we've adopted. So we don't know. There's a lot we don't know, but there are these intriguing hints here. So overall, this mixture of characteristics or components that are differentially consistent with monogamous mating systems suggests that there might be ongoing social transitions in mountain gorillas that are interesting and important to study now as a potential comparative model for seeing different routes by which these components of monogamous mating can co-occur or not co-occur. So there's a lot of smoke, right? Is there a fire? Well, we'll see. I think that there is. I think there's enough hints out there that there's something going on, but we have to do the work to figure it out. So this project that I'm working on with Stacy Rosenbaum now, we will collect a lot of this data one way or another, but the outcome of some grant proposals depends on what exactly all we will be able to collect. But using a combination of this long-term data set, this archival data that we have access to through the FOSSI Fund, and then newly collected regimens of data collection, we want to know, okay, well, are there identity-based preferential relationships between male-female gorilla pairs that exist independently of these transient states that we already know predicted? Things like, independent of transient states such as female parity or receptivity. And then also, it's not an either-or thing, right? It's not either totally undifferentiated or extremely strongly differentiated. There's a wide middle ground. So the next part of that question is, how differentiated are these relationships, right? If you go back to this plot here, do you see like it looks pretty flat, like the Gutengara's plot there, where there's maybe a slight preference for one, but it's a pretty flat distribution, or do you see really high skew, where one male is highly favored over all others? Both of those are possible. And next, we are interested in seeing if there are different dimensions of mountain gorilla bond quality, right? Instead of saying there's just like one unitary factor of like a strong bond versus a weak bond, I think it's plausible that there are different components that make up this bond, right? This is kind of something that is, of course, a no-duh statement. If you're a relationship scientist and a psychologist in humans, obviously there are different aspects of romantic relationship quality. There's been a little bit of work in chimpanzees showing that there are these sort of overlapping but distinguishable characteristics of bond quality in chimpanzee pairs. So we want to kind of build upon that and see if we can find different aspects of mountain gorilla bond quality. And then because of our access to the long-term data, we have the ability to ask what consequences, if any, these bonds and the strength of the bonds have for fitness outcomes, right? Do more strongly bonded pairs have a higher likelihood of having repeat paternities together, or repeat offspring together, rather? Does bond quality predict lifetime reproductive success? Does it predict better health of the infants that result from it, and then better health of those infants when they themselves become adults? Because now we have, generational data that we can look at there. All that stuff is stuff we want to look at, possibly more, possibly less. One of the things that I'd be happy to talk about in the Q&A session is ideas that you have or questions that you have about mountain gorilla land and what might be interesting and feasible to test with this both long-term data set and new data collection regimen. But stay tuned on that. So I'll close with just a couple slides. The first is summarizing the point that hopefully you now agree with me, or at least by some aspect of the fact that these non-traditional models have a lot of promise, right? We know really very little about a lot of our primate cousins. These examples are just two of them. So studying lemurs or mountain gorillas or some other neglected group, but it doesn't just serve to fill in a gap in our knowledge. It also presents an opportunity to develop an animal model for understanding diverse or atypical social arrangements, has implications for the generality of previously proposed biological mechanisms or models of social evolution. And here I just gave an example of two different clades where the findings have implications for understanding the evolution of primate social systems. And there are so many fundamental questions left unasked, in my opinion, right? This is really just scratching the surface. Knowing what underlies a species' distinctive behavior, sociality or physiology, do mechanisms discovered in these models apply to other species? The answer is we don't know, right? So that's where I'll close, with an exhortation to everyone to think about snark hunting in their own research. So I've given examples in my talk that draw from my field of sort of behavioral endocrinology and primatology, but I bet everyone here can think of similar instances in their own research where there's a need to broaden the models studied if we want to understand both generalizable principles and disparate features of whatever you're interested in. So here's one last illustration from the book, from Carol's book. It's a map that one of the main characters, the bellman, provides to the rest of the crew towards the beginning of the story as a guide for sailing to the unknown island where the snarks live. The absurd part, of course, is that the map's blank and seemingly useless, right? But the bellman thinks otherwise. He says, quote, "Other maps are such shapes with their islands and capes, but we've got our brave captain to thank that he's brought us the best, a perfect and absolute blank." Okay, so to be clear, the analogy here is not perfect. I don't think we should be sailing blind as scientists or anything like that. But I do think there's some value in seeing an unfilled map not as a liability, but rather as an opportunity. That's where I'll stop. Thanks a lot for your time and attention.